Okay, AP Bio. So we're going to take a look at gene expression now, and specifically we're going to look at different types of genes, how they're regulated, how they're controlled, and the mechanisms behind ensuring that we produce protein when we want to and when we need to. So just a quick recap what genes are. Remember, genes are sequences of nucleotides. They determine the sequence of amino acids and proteins. We have two types of genes. We have regulatory genes, which control transcription. They're going to regulate whether or not a gene gets turned on. So a lot of these code for regulatory proteins, activator proteins, transcription factors, and some of them also code for RNA. There's interfering RNA, which is coded for by regulatory genes, and that's going to help silence the expression of certain proteins. And then we have structural genes, which determine the actual physical nature of a protein. They determine the amino acid sequence and how those amino acids come together. And ultimately, they're going to control what the amino acid sequences look like. Okay, so structural genes are going to be for genes like hair, skin, nails, enzymes, pigmentation, biological structures like that. Okay, and so when we look at how genes are regulated, the simplest one to look at is the prokaryotic gene mechanism. And prokaryotes regulate their genes through a system known as the operon. Okay, operons are a group of specific genes within the DNA that control transcription of the RNA. Right, and they're going to control protein synthesis. Okay, and there are four parts to operons. Okay, the first part is the regulatory gene, which is a gene located somewhat upstream from the actual gene itself, the structural gene. And this codes for one of two things, either a repressor protein or an activator protein. Okay, and they do exactly what they're named for. Repressors repress. Okay, so they're going to be an off switch, and the activator protein is an on switch that's going to start transcription and promote the production of protein at some point. Okay, motors, region of DNA where the RNA polymerase binds, that's your start codon. So that's going to be your AUG start codon. For RNA, it's AUG. But that's simply where RNA has the ability to bind and start the process of transcription, reading the DNA to make messenger RNA. Right next to that is the operator, which is the actual on-off switch. And the operator is related to the regulatory gene because the regulatory gene produces the protein that binds to the operator. Okay, so that repressor protein or activator protein is going to interact with that operator, and that's going to control the transcription of genes. Okay, and then finally, the structural gene, that's the fourth part, and that's the part of the gene that actually codes for the protein that we want to produce. Okay, so with these operons, they can be inducible or repressible. Okay, inducible means it's off all the time. We have to turn it on, and we only turn it on when we need the product of that particular gene. This is going to require an activator to remove the repressor protein. Now, this activator is also known as a co-repressor or a co-activator. Okay, and what will happen in the case of the lac operon is the lac operon is off all the time, and it's only activated in the presence of lactose. Okay, lactose is the sugar that needs to get digested by lactase. So the lac operon codes for lactase. Okay, and so that's only activated whenever lactose is present. So we don't want to always produce lactase unless it's absolutely necessary. Otherwise, it's a waste of energy and resources. Repressible genes are on all the time, so they're always producing their gene product, and they only get turned off for short periods of time when either there's an excess of that product or if it's freely available in the environment. The trip operon is an example of this. The trip operon produces tryptophan. It's an essential amino acid for bacteria. Okay, and so if there isn't enough tryptophan in the environment, bacteria can manufacture it themselves using their trip operon. Okay, so let's take a look at how these work. So I'm going to jump to the animations. We'll take a look at the trip operon first. And again, the trip operon is set up in such a way that we have two sections to it. We have the regulatory gene, which is several 100,000 base pairs up from the main gene. Okay, this gene codes for the repressor protein. Okay, and then we have our promoter for the trip operon. This is the operator, and then these are the genes that code for tryptophan. Okay, and so with this trip operon, when we produce the repressor is produced in the inactive state. Okay, and so that repressor protein can't attach to the operator. So as a result, the RNA polymerase has the ability to just copy the gene. okay? And when we look at this repressor protein, we can see that it's in a form that's inactive, but it has this little opening on here, okay? That's a regulatory site, so that a substance can attach to this and change its shape. So this tripoperon, um, rather this regulatory protein of the tripoperon is allosteric, okay? It exists in two forms, all right? Right now it's in the inactive form. And so in the inactive state, we permit RNA polymerase to copy the genes that code for mRNA. This mRNA is translated into the proteins that form the enzymes of the tryptophan pathway, and this is going to produce tryptophan. Now, as the tryptophan levels increase, eventually they're going to reach a critical point where some of that tryptophan is going to attach to this repressor protein. Now, remember, this repressor is inactive right now, so it's permitting transcription. But when enough tryptophan builds up, it binds to this and acts like a co-repressor, and it's going to change its shape. Now the repressor is activated. It's allosterically active. It binds to the operator, and it's going to inhibit RNA polymerase from moving, and the gene is off. As a result, because of this, we do not make messenger RNA, we do not make the proteins, and the production of tryptophan is inhibited. Now, once the tryptophan levels come down again, there becomes a need to make more, and we remove the repressor, and we're now able to produce tryptophan again. Okay, and here we go. There's transcription, translation, and we make our tryptophan. Okay? All right, now we're going to take a look at our lac operon. Okay, and lac is the opposite. Lac is inducible. So we start off here with our lac operon. Okay, and again, same scenario here. We have a regulatory gene here indicated by the gene I. Okay, there's a promoter here that promotes it. And then we have a promoter for our lac operon. We have the operator and we have the lac genes. We have the lac operon genes of Z, Y, A. These genes each code for proteins that come together to form lactase. Okay, so when we produce our lac repressor, it's in the active state and it's able to bind to the operator. It recognizes the specific gene sequence and it attaches to the DNA. Okay, so when that lac repressor binds to the DNA, it turns off that gene. This is the way the lac operon is all the time. Okay, so those structural genes of the lac operon are not going to be transcribed. Okay, and again, the reason for that is because that lac repressor is going to block transcription, and it's going to prevent the RNA polymerase from moving along the DNA. So here we go. Okay, there's our repressor, it binds. Okay, and the RNA polymerase will attach to the promoter, and it's unable to advance. It can't copy the genes to produce lactase. So without RNA, we do not make any protein. So in this scenario, the lac operon is repressed. It's considered to be off. Now, if we introduce lactose, okay, lactose we know is a disaccharide. It's also known as allolactose. It actually goes through a slight transformation when it enters the cell and it becomes allolactose. So this has the ability to attach to the regulatory site on the lac repressor. Okay, and so watch what happens when it attaches to this regulatory site. It's going to alter the structure of the protein, and it inactivates it. Okay, so now this is allosterically inactivated. So if we want to try to turn this off, we can. Okay, and so because of this, the operator is no longer blocked, and RNA polymerase is now free to move along the DNA and transcribe the DNA into a strand of messenger RNA. Okay, and that transcript gets translated at the ribosome into the three proteins that together form lactase. So these three enzymes come together and they form lactase, and that's going to permit lactase to digest the lactose. Okay, once that lactose is digested, we go back to the original scenario that we had, where without any lactose present, the repressor protein can reattach and block transcription. Okay, and then the polymerase can't move, and we've now turned it off again. What this does, this acts like this is negative feedback. And so what this does is this prevents overproduction of lactase so that we only make, or the bacterial cells, I should say, only make the amount of lactase required to break down the lactose. Once the lactose is gone, we shut down lactase production so that we inhibit overproduction, and it helps maintain an efficient mechanism of metabolism. Okay, so in both cases, we're running this negative feedback loop. Okay, if it was positive feedback, what you would see is as you produced more lactase, that would trigger additional transcription, which would trigger additional lactase production. But we don't see that. The more lactase you make, the quicker you're going to digest the lactose, which means you're going to quickly inhibit and shut down lactase production. Okay, it's known as negative feedback because your initial response to the presence of lactose is to make lactase. What that does is it shuts down lactase production. Okay, and that's why it's negative feedback, because the response that you're initiating is going to wind up producing something that shuts down that initial response. The same thing happens with the trip operon. Okay, your initial response is to, the trip operon's on all the time. You're making a ton of tryptophan. If there's too much tryptophan, you shut down the repressor. Okay, you shut down the gene by activating the repressor. Okay, and then that turns off your tryptophan production. If it was positive feedback, the more tryptophan you make, that would signal you to produce even more enzymes to make even more tryptophan, and so it would just continually build. Okay, I hope that makes sense regarding negative feedback and positive feedback. Okay, so let's go back to the PowerPoint, and we want to take a look at eukaryotes. And eukaryotes, much more complicated. They utilize a wide range of factors. They utilize transcription factors that increase the rate of gene transcription. Um, they utilize promoters, mediator proteins, enhancers, which are activators, and acetylation. Okay, acetylation is a simple thing. If you add acetyl groups to the DNA, it increases the chance that you're going to transcribe it. So the RNA polymerase is more likely to bind and copy that gene. Enhancers are proteins that basically act like homing beacons. They increase the rate of transcription. They generally bind to switches and then attach to the mediator. Okay, and that's going to trigger transcription. Okay, um, and so when we look specifically at these, this is an example we looked at, the regulatory switch in the stickleback fish. Okay, and this was from that PIDX1 activity we did. Okay, the gene right here is the PIDX1 gene. This is the protein coding region. Okay, here's our RNA polymerase bound to the promoter. Okay, now the general transcription factors bind to the RNA polymerase, and they permit mediators to also attach. Okay, what these transcription factors do is they hold the polymerase in place. They prevent it from moving. Okay, until a proper signal is achieved. The mediators are going to bridge the gap between these activators and the general transcription factors. Okay, the gene does not get copied until an activator attaches. The activator is going to activate gene transcription. That is coded for by switches. So there are all these regulatory switches here that each produce a different activator protein. When the activator connects to the mediator, it changes the structure of the mediator, which then changes the structure of the transcription factors, and the RNA polymerase is permitted to leave the promoter and copy the gene. If you're missing a regulatory switch, you do not activate the gene. The mediators and the general transcription factors will just sit there for an eternity and wait for a signal, but without a regulatory switch, the signal never comes. And that's what happened with our little stickleback fish, okay? There were two forms of the stickleback, okay? One was the marine form, which has the pelvic spine, and the other one was the freshwater form without the pelvic spine. And when they did their analysis on these two fish, they noted that the marine fish had the switch for all three of these, the pituitary, the jaw, and the pelvic switch. And they produced a pelvic switch activator that would bind and then trigger the mediators to activate. Okay. With the stickleback fish that didn't have the spine on the pelvis, there is no switch. Therefore, there is no activation of PIDX1. And so without that, this activator switch doesn't bind and it can't interact with the polymerase. Okay. However, what they did, if you remember from the movie, they spliced a pelvic switch back in and then the fish produced offspring, and the fish itself had the spines. So by adding the gene back in, they were able to do that. But what was interesting is this particular mutation is a very common mutation. It was a deletion of the entire switch. And so that rendered these fish to not have that spine. And it's a common mutation, but in the marine environment, you need the spine to survive. So any fish that were born without a spine were quickly eaten. So they were eliminated from the population. However, the fish without the spine in the freshwater population, they now had the advantage, and that's why they were able to survive. Okay. Um, Now we look at humans, and we'll take a look at this. And this is an example in humans where we have gene regulation by hormones. And here's the estrogen molecule. Estrogen is a lipid hormone, okay, just steroid hormone. So it can pass right through the cell membrane, goes into the cytoplasm, and then it goes and meets up with a transcription factor, which is the estrogen receptor, right? So estrogen binds to this receptor, activates it, and then together, this nuclear hormone receptor with its hormone binds to the DNA at the promoter, and it's going to trigger gene activation. Okay, what this will do is trigger messenger RNA to be produced, which then produces specific proteins that affect cell behavior. In particular, one of these proteins regulates mitosis. Okay, now what may happen in some women is, particularly in the cells in the breast, they will have an overproduction of estrogen receptors. And this results in estrogen signaling the cell to divide more often, which can lead to additional mitosis, which leads to tumor growth. And so estrogen dependent breast cancers are specifically indicated by a high concentration of estrogen receptors within the cell. So that indicates that not that the body's making extra estrogen, but it's making extra receptors, which means whatever estrogen is around is binding to more of these receptors. And with more receptors, you get more signals to divide. Okay. And so what scientists did is they developed a hormone, not even a hormone, a drug treatment called tamoxifen. Tamoxifen blocks the receptor. What it technically does is it binds allosterically to another location on the receptor protein, and it changes its shape. So estrogen can't attach, right? So without estrogen, we do not have the ability to bind to the DNA. So we shut down gene activation, which means we shut down uh, gene transcription and translation to protein, and we don't make those proteins anymore. Okay, estrogen is still being produced; it's just not able to bind to this particular receptor, which happens to trigger cell division. So estrogen is still working; it's just not working on this particular receptor that is changing cell behavior, such as cell proliferation. Okay. All right. Now we can also inhibit transcription. Methylation is the most common way to shut down transcription. Um, if you add methyl groups to DNA, they like to attach themselves to cytosines that reside in CG doublets. And what that does is it triggers the DNA to coil up. And so it impedes the transcription of the DNA, so you can't copy the genes. If you demethylate, you open up the DNA, and by removing the methyl groups, now we expose the genes, and these areas that were once coiled up can now be transcribed, okay? And now we can express those genes, okay? There are lots of things that can affect methylation. When we do methylation, we're silencing, right? We're condensing the chromosome, and we're causing those genes to be turned off. So epigenetics talks about modifying the DNA by altering gene activity. And usually this occurs through methylation. Okay, one of the most common effects of epigenetics is BPA, which causes demethylation. Right, this activates genes that trigger obesity, and it also activates genes that trigger diabetes and developmental problems. It's an estrogen-like substance. It's not an estrogen, but it does affect cell growth and cell differentiation. And the thing with epigenetics is we can see this running in multiple generations, okay, and it has an impact further down the line. And then the other thing is RNAi, okay, interfering RNA. Basically, what RNAi does, I want to see if I can draw on this. So since we're towards the end, let me get fancy here. Let's see. Yeah, let's use this pen. All right. So what'll happen is you'll have a ribosome. Okay. Okay, so this is a ribosome. Okay, and it's moving along, right? It's reading 5' to 3', and it can only read single-stranded RNA. Now, what happens is siRNA stands for small interfering RNA. It's a small sequence of RNA that perfectly binds and complementary base pairs with messenger RNA. And what this does is it blocks the ribosome from completing its job. So when the ribosome gets to this point, it stops and it falls off. And what you're left with is a truncated protein. Okay, you don't make a complete protein. Okay, and so what this is doing is this is post-transcriptional modification. We are inhibiting gene transcription. We are inhibiting gene expression rather by modifying the protein before it's completely done. And by modifying it, what we're basically doing is we're not allowing it to be completed. Okay, so it's like a roadblock. And um, miRNA, microRNAs, do the same thing, but the difference is they only base pair partially. So you get what's called a seed that tends to bind in a small section, but ultimately it does the same thing. And once this ribosome, same thing, gets here, it can't copy anymore. The other thing that these both do is they both trigger enzymatic hydrolysis. So from this point on, the RNA gets destroyed by enzymes. Okay, and that's because these both bring in components that signal degradation enzymes to get in there and start breaking apart the RNA. So basically, what an RNAi does is it interferes with the translation process. Okay, this occurs after we've made the messenger RNA and the ribosome is reading it and putting together the polypeptide, and it results in a truncated or shortened polypeptide. Okay, that's it for gene expression. It's a lot of stuff. It's a lot to digest. I hope that this video makes it really clear for you guys. And ask me questions. We're getting there. We're getting close to the end, guys. Next week's the last week, and then we have the exam. So hang tight. This is some tough stuff, but just bear with it. Go back through it, look over it again, and hang in there. Okay. I'll talk to you guys real soon. Take care. Bye.